Hey, Audrey.
Hi, how are you?
I'm doing all right. How are you?
I'm good. Definitely a Monday though. It's chilly outside.
The weather did turn on us, and I'm a little worried about what that's going to do to lab, but hopefully it's not going to affect things too much. Um, Dr. Judd and I are feverishly writing up protocols for lab this week, so we're feeling a little under the gun, even if you guys might not be.
Hopefully it's all good.
Yeah, we'll see tomorrow morning at 7:45 how things went. Angelina, were you in Mamma Mia or did you go to see Mamma Mia?
I went to go see it.
Okay. A high school production?
Yeah,
How was it?
it was decent.
Decent? That's not a resounding review.
I mean,
I've never.
my high school did a good job of recreating the show.
Okay.
I felt like they could have done better, but it was still decent.
Don't we all feel like we could have done better at times?
Let's see.
Oh, isn't it?
Hello.
Hey, Dr. Judd.
Just trying to get something posted on Moodle here for you. All the slides for today are now up. And I want to put like a announcement or something. That's what I'll do.
Dr. Judd, do you want me to start working on breakout rooms?
Yes, that would be great. Okay, so welcome everybody. Today we're going to do two things. We're going to talk about your summary of the methods and results by Henry et al. And then once we've gotten through that, we're going to start on a new topic that's going to take us through the rest of the week. This is going to be genetics and inheritance, principles of inheritance. So that is now up. We've just about finished with all the exams, so you all should start getting your getting results back on that shortly. But we're going to save that till after today's lecture so that we can focus on this stuff. You've got labs coming up this week, so you'll expect to get some protocols, some lessons from us on how exactly you're going to collect your data and what kinds of analyses you're going to do. For those of you who are coming in on Tuesday and then I guess later on Thursday, make sure you've finished your homework assignments for lab, thinking about data management and thinking about the golden rods. Okay. So it looks like most of you have submitted the methods and results summaries up here. We're going to get you back feed comments on both your introduction summaries and on your methods and results summaries sometime in the next several days. And you'll be able to use that information, use that feedback and the class discussions, which are recorded, to improve and make your final summary as strong as it can be. So you can look for those. I'm going to try to get them with all the introduction stuff tomorrow. So see if I can get that back to you shortly. Okay. So what we're going to do is we're going to split you up into your small groups, and we want you to share a little bit about your summaries of methods and results. And to guide you in that sharing discussion, I have a series of questions that I'd like you all to go through and talk about. I've posted them on Moodle, but I'll also post them in the chat here. And there's several, maybe there's a little bit of overlap. It's more important that each of you contribute a little bit to the discussion in your group than it is that you have a full. So if you see things that seem a little bit redundant, just make sure that you return to those points if there is some redundancy. Okay, so
Nathan,
yeah,
I made you the host so that you can move from room to room, but
oh,
I hope it didn't. I got everybody assigned to breakout rooms, so I hope it didn't mess that up.
okay. Let me see if I can copy this into the chat before I go anywhere so you guys can see it. But just to be safe, I did put the questions on Moodle for today. Not getting the chat copied over. Why is my copy not working? Command C. Okay, I'm going to ask you guys to look at Moodle for the questions because I'm not able to copy and paste this for some reason. The problem is there. Can I get a thumbs up if you guys can see the questions on Moodle? It begins with recall the big picture. Okay, good. All right. Okay, let's see if we can get you in your breakout rooms. Okay.
Are they all there?
They should all be getting invitations right now.
Okay, cool. There they go.
Room 1 is only one person this time.
Oh, really? It should be Tavares, Audrey, Brandon, Cody, and Joseph.
Yeah, I didn't get an invitation for that one.
Me neither.
Me
I
neither.
haven't gotten an
Oh, I
invitation.
see.
Me either.
Okay,
Dr.
I
Judd
see a
now has control, so.
Yeah. It says assign to.
I can just tell you, I'm looking at it right in front of me. So
Okay.
Tavares, Audrey.
Yep. Audrey. Who else?
Brandon, Cody, and Joseph.
Okay. And then I have 7 others that didn't get the assignment.
Connor is in group 4.
Yep. Got it. Alex.
Riley is in group. Okay, let's just do group 2. Riley and Alex are in group 2.
Got it.
Group 3. I think everybody is in group 3 who's supposed to be in. Ryan is in group 4.
Okay.
Oh,
Casey.
Casey is in group 3.
And lastly, Michaela.
And Michaela is in group 6.
Got it. All right. Partial success, partial bug.
Yeah, and you can stick me wherever you want.
Okay. See.
And I will live out my life stuck away in that hole.
Let's do. I just picked one.
All right.
And then for what did they measure? I said like beak length and depth and width, I think. And then they were trying to see like how it was different on the different islands that had the different population growth.
You guys are super not talkative.
Okay. I guess what were the like salient features? What did you all say for that of the methods?
No, for the things that like you couldn't leave out, I like talked about like you kind of had to say whether they found like which ones were unimodal, which ones were bimodal, because that was kind of important to like what they like how they were able to determine like the impact of the humans in the area.
Yeah, I pretty much put the same thing.
Were you guys confused by all the different ways that they tried to measure things like bimodality?
A little bit, yeah.
Okay, what confused you?
I don't know, like part of it made sense, but I don't know, like the whole thing was just like a little confusing. Like I guess I don't know exactly what, but I was still confused.
Okay. So one of the tricks of understanding what you're reading is figuring out what's confusing, like identifying it, but then figuring out what is confusing about it. So we went through some of the ways in which they're measuring bimodality in class last week, right? How did they do that?
We talked about like the PC1 or whatever in class.
Okay, so that's not really how you measure bimodality, though. We showed you a way of visualizing it, right? Oh, you guys are really quiet now.
We did like the graphs with like the regular frequency versus like the cumulative frequency to show like where like sometimes it was like more like higher than expected and sometimes it was lower than expected.
Yeah, so one way is by making those things called QQ plots, which are a plot of the expected cumulative frequency on the x-axis and the observed cumulative frequency on the y-axis. And so that's one way of doing it. Now, that's a very qualitative way of measuring bimodality, but so they use some more sophisticated ways of measuring bimodality that are mentioned in the paper. And so when you look at the paper, they talk about these information indices. And those are just quantitative ways of basically examining the same kinds of deviations. Was having gone through that discussion of bimodality in class useful to you or not?
I think it was.
So you don't probably understand anything about Akaike's information criterion, which you shouldn't be expected to know about it, but this is a method for essentially quantifying the shape of a normal curve, and they did the same criterion on the shape of a bimodal curve and essentially subtracted those two measures from one another, and it gave them a range of values, and those range of values that they looked at indicated whether something was unimodal or bimodal or kind of in the middle of things. And so when you look at figure 4, there's a lot of information there. You can see the data plotted, but then you also see delta and W and the word 2 or 2 equals 1 or 1 equals 2, etc. What is the delta?
Isn't it like like it says like it represents like the change for like the single normal distribution minus like the 2?
Yeah, so it's this information criterion, and it's just the difference between what it would be for a unimodal versus a bimodal distribution. So that's the thing. So you can look back at these ranges on page 1889, -8 to -5, -5 to 5, 5 to 8, and above 8. And those things should correspond to the decisions that are talked about, whether it's 2 versus 1 or 1 equals 2, et cetera, that refers to those ranges. So for example, Academy Bay 1968, that delta value is 58.2, which is way above a value of 8, which would indicate strong bimodality, and that's why they say that this is more likely to be two distributions rather than one. Academy Bay in 1973 has a delta of 1.2, which is kind of there in the middle, where it could be just as easily coming from 2 different distributions or one single distribution. But one of the things you notice there is they don't have very much data from 1973. So the amount of power that they have for determining whether it's bimodal or unimodal isn't very high. They don't have a lot of statistical power to make that decision. So what you see in the figure should reflect what you see in their treatment of the methods on page 1889. Continue chatting amongst yourselves. I'm stuck with you for the whole time because I can't move from room to room. Have you guys exhausted all the questions? You guys are killing me, you know that, right? What questions do you guys still have to address in here?
We were to do the space for time one, but I was trying to like figure out what that like was.
Okay. Does anybody else know what that is? Look up on your web browser, space for time substitution.
So is it basically just looking at how like a certain area changes over time?
So if you're interested in a long-term change, you can either measure something at time X and then wait a long period of time and then measure that same plot at time Y, some distant time in the future. But if you're going to do that, you have to live long enough for the time to have passed that you're interested in. What you do in a space for time thing is, rather than waiting for time to pass on a single plot, what can you do?
I guess like look at a different plot that like has changed.
Yeah, so in this case, they are looking at Academy Bay. It was one thing in 1964, it was doing something else in 1973, and it was doing something else in 2004. So at Academy Bay, we have this trajectory of time where we have measures at these different time periods. What do we have at El Garapatero?
That one like did not change very much at all.
We only have measures from El Garapatero when? We only have El Garapatero in 2003, 2004, and 2005. We don't have measures from there in 1964 and measures from there in 1973. So, or is it 64? Am I remembering the dates right? Yeah, 64, 68, 73. So in El Garapatero, we don't have older data, but presumably what's not happening at El Garapatero?
A lot of change.
What would be driving change at El Garapatero?
Well, wasn't it like people, but there's not as many like people there?
Yeah, so there's no human increase here. But there has been human increase here. So time has proceeded in this trajectory. So as a result of that, because human increase has changed here, there's no way of knowing if 2004 data from Academy Bay is due to human increase or if it's due to some other change going on in the environment. Maybe it's climate change that is having an effect. So how can we kind of go back in time in a way to what Academy Bay would look like in 2004 if we could get on a different trajectory where Academy Bay wasn't the site of a town, and discover, well, what would Academy Bay be like in 2004 if not for all those people? Well, we don't have a time machine. And we also, in both of these places, if there is anything going on with something like climate change, it's presumably affecting both areas the same. The only difference between these two areas is the presence of humans or the lack of presence of humans. And so these dates at El Garapatero is basically what Academy Bay should look like in the 2000s, if not for humans. So we use spatial variation so that we don't have to build a time machine, essentially. Or, this is only one area here, and it's an area where humans have increased. There are all sorts of things that could lead to Academy Bay changing over time. Human increase is just one of them. Well, if these broader things could be affecting El Garapatero and Academy Bay the same, then having El Garapatero to compare to tells us to what degree change is happening because of something other than humans. If you saw the same amount of change, if you saw the same types of changes at El Garapatero, what would you conclude?
Something other than humans.
Yeah, you would conclude that it's something other than humans going on because humans occur at one place and not at the other place. And so, once again, you're using spatial variation to stand in for this change or lack of change due to human population size increases over time. So that's what a space for time experiment does. It allows you to look at change over long periods of time without having to live forever. What other questions have you not addressed so far?
We still do the last three. It's like, what did they measure and why?
Okay. So what did you measure and why? Yeah, and you guys answered that or not?
No, we have not.
Okay. Because you guys were talking about analysis of the data when I popped in.
Yeah, we weren't directly talking about it, but kind of like half already talked about it.
Yeah, because that's kind of inherent in talking about what they did with the data. So address that.
So, because they measured the beak length, width, and depth.
Okay.
But then they also went back and just found data from what other people had measured, and in some cases it was measured slightly differently, or the beak, I think, width wasn't measured in all of them.
Mm-hmm. And then how did they deal with the fact that different people at different times measured birds in different ways?
That's when they went to the principal component analysis, because it was an easier way to combine everything. And then they also did the principal component analysis without beak width to kind of make up for some people not measuring that.
Yeah, so one of the nice things about doing principal component analysis is you can get a measure of kind of relative beak size that is kind of independent of slight differences in how it's measured. You have 57 seconds to talk about the results.
I guess basically they just determined that in Academy Bay, it was the most change going from bimodal to unimodal, right? And then in both Barrero Bay and El Garapatero, it stayed more of the same.
Yeah, so and Barrero Bay was a weird place because they didn't really have very many large birds. So there's no bimodality there, but not because of any change. It's just a reduced distribution of the birds in terms of size distribution. And El Garapatero is still bimodal after all this time. And why are these birds at Academy Bay going unimodal? What's the main driver? What are humans primarily doing?
Okay, that looks like we got pretty much everybody back. So who can help me distinguish between the big picture question that we covered when we talked about the introduction and what specific question they were after in terms of the methods they used and the results they obtained? Who wants to take the first half of that? Remind us what the overarching question was about selection and evolution and humans.
I think they talked about the question was like, does human interference affect biodimality? Yeah.
Yeah, so you've actually got the two kind of together there that I was going to try to tease apart a little bit. So do humans affect diversification or evolution of species at all? And specifically, can they interrupt diversification? But bimodality is the answer to the second part. What specifically about bimodality were they interested in investigating here?
Weren't they researching how like it was bimodal until like people started showing up on the island and then it went kind of unimodal?
Yeah. Yep. So that's what they wanted to see if they could detect.
Yeah.
Yeah. Good. And what was their approach to checking for that?
Like,
Well, I guess first of all, is this a mensurative or a manipulative experiment? Did they design things and manipulate, or did they just find different scenarios, observe them, and compare the results under the different scenarios?
they found different scenarios. So like they went into a population where there was a lot of people and like captured the finches or whatever and measured them and then released them, and then went into a part of the island where there was no people and then did the exact same thing.
Yeah.
And collected data like that.
Anybody want to add to that? Did Henry all do all of the collecting of all the finch measurement data at all the different sites?
No.
Good. They were, where did some of the data come from?
They like took it from people who had like measured the different like beak like stuff in the past.
Yeah, good. And what was the value of measuring at places like El Grapatero? If they're interested in what happened at Academy Bay, what's the point of doing all this measurement at El Grapatero?
It was like a control group.
Yeah, good. That's cat. So a lot of things happen at Academy Bay besides just human population increasing, right? All kinds of things happen when time passes. So comparing before and after just Academy Bay would just tell you that something happened. Wouldn't necessarily tell you that a change was because of human population increase. But you can kind of isolate that with these control groups by looking at other areas. Oftentimes, when you have a historical phenomenon like this, your sample size is kind of one, right? It happened at Academy Bay once. So how do you increase confidence that your explanation, your hypothesis, is actually in fact what happened, what explains that historical event? So what were they measuring? Or what were they trying to measure when they captured the birds?
Didn't it say like beak length, width, and depth?
Yeah, so that's the raw data.
Yeah.
Can you remind me why they used PCA if they had that raw data?
Me or someone else?
You can take it if you want.
They,
I did.
oh,
He's passing it off. Who was that speaking up? I just heard.
it was me. So they calculate the PCA to find the beak shape, like to find a new way to measure the beak shape
Yeah.
from the 3 data they measured.
Good. Yeah. So beak shape is one of the things that influences the food that the finches have access to. Beak length by itself, is related to that, but it's not the whole picture. So they wanted to look at the distribution of beak shape. And that's why they used the PCA. So good. Really good. Okay. Who wants to tell me about this methods of analyzing their data? If they're interested in bimodality, they actually had a lot of analyses, right? Why was there so much analyses? Why did they have different methods? Why did they have so many graphs? What's going on there? Well, first of all, if we look at the bar charts, why were there so many? There was more than just 3, right? 1 for each site.
They did it for like each individual sample.
Yeah, and why did they do that again?
Each sample wasn't measured exactly the same. Some researchers didn't measure the width.
Yeah, so this was nice to have access to this historical data, but the people collecting data in the 1960s didn't know that Henry et al. Were going to write this paper, right? So the methods for collecting data weren't standardized yet for this analysis. So good. And what about the results? What did they find?
They found that through time the bimodality is like getting less strong. Is that
Yeah, it can be a little bit awkward to phrase that, right? But there's a shift from strong bimodality to unimodality, right? So you can try to put that in your own words, but I understand, I've read enough of these here, it can be kind of awkward on how to say that exactly. But where did they see that shift?
Academy Bay.
Yeah, good. And did they see it anywhere else? Or what did they see elsewhere?
In El Grapatero, it like stayed bimodal, and then in Barrero Bay, it stayed unimodal, but that was just because they didn't have like many of the birds with the large beaks.
Right. So there was less variation just because of what happened to be the case at Barrero Bay. So you wouldn't necessarily expect to see a change in the distribution if there wasn't enough variation anyway. So they talk about the concept of the coefficient of variation. And that's a way of seeing how wide the distribution is, whether it's bimodal or unimodal. It just tells you kind of how wide it is. So you can see that at Barrero Bay, the distribution of beak sizes is much narrower than elsewhere. What about these different ways that they tried to assess bimodality? Any questions about those? You guys see my whiteboard now? One of the ways that they went at this, how much time? Yeah, okay. One of the ways they went at this was a kind of a visual way to assess bimodality. So imagine that you've got a normal distribution shaped like this, and you want to compare it to a highly skewed distribution like this. So A, B. If we put the normal distribution down here, sorry, I didn't realize it was sideways. Come on. Let me try that again without the sideways business. All right. So A, B. As you go along this distribution in A, at the midpoint here, you've got half of your observations on one side and half on the other, right? But in this skewed distribution, you've got half of your observations are maybe on this side and the other half are way over there. So if you mark the halfway point of distribution A here, and then you ask where is the halfway point for distribution B, it's going to be maybe down here, right? Before you get to the halfway point of the range from 0 to many. So in a distribution that's skewed like this, what you'll see is that as you accumulate observations in A, initially you accumulate them very fast in B, and then later you get the rest of them. So you get this curve to the line. If we had the distribution go the other way, then it might take us a really long time to get to the halfway point. And so you get this kind of a shape. If you've got a bimodal distribution, and it's got that same range, then initially you pick up observations more quickly in the bimodal distribution than you would in the equivalent portion of the unimodal. But then you get to the halfway point, it's still a halfway point, so it's got to cross again. And if you had two normals, you'd get this yellow one. So if you get this S shape, which I think I've actually drawn reversed from what they present, my S is upside down, then that means that you've got a bimodal distribution. But the key here is that this is kind of a visual judgment call. There's not some t-table that you look up and say, oh, it's bimodal in this case. Now, there's a nice animation on how to think about these things on Moodle. So we do want you to understand what these cumulative frequency distributions mean and how they work. I just wanted to give you another chance to think about that. Okay. Let's see. I had one more question for you about this, and then so let's relate the final result back to the big picture question. What did they find in terms of what they can say about whether humans affect diversification? Did they convince you, or do you think there's more that it would take to convince you? This question will be the heart, hopefully, will be near the heart of your final paragraph in your literature summary. Was their study convincing, and what additional things could you do to check to see if their interpretation of the data is correct? What future studies could you do? While you marinate on that for a moment, want to give you one more term, and then we'll switch over to genetics. We talked about the horned lizards, a study that you guys all seem to understand really well. A lot of you use that as your example on the exam. We saw directional selection. Does anyone know what I mean by directional selection in the context of the horned lizards? What kinds of lizards were more likely to get killed?
Parental.
Say that again.
The parental.
Are you talking about the? Sorry, I just didn't hear if you said parental or parietal.
The second word is what I meant to say.
Oh, okay. Yeah, okay. So, and what kinds of parietal horns were a better defense against shrikes?
Covered the back of the necks.
And what was the variation they observed in those horns that cover the back of their necks?
They were longer.
Yeah, good, right? So the shorter ones were more likely to get eaten and killed by a shrike, and therefore not produce offspring for the next generation or have lower fitness, right? So because you got this bias against survival of short-horned lizards and for ones that have longer horns relative to their head length, what we see is the response to selection, the response to having those short-horned lizards removed from the population is a directional shift. So a change in the average horn length in the past, presumably to a longer average horn length in future generations. In the finches, that is not what we're talking about. In the finches, before humans showed up, there was that bimodal distribution of the Academy Bay, and we still see it at Agorapatero. That's weird, really drawn. Does anyone know what we might call the kind of selection that would produce this shape? This is your new vocab word. It should be in the paper, but I don't know that we've said it in lecture yet. Because there are abundant resources that are easy for small-beaked birds to get at, and resources that it's easier for large-beaked birds or more robust beaked birds to get at, the ones in the middle have this lower fitness. So you can think of it as there's selection against having that intermediate beak shape, or intermediate beak size, excuse me. So the plants and the seed availability, the food availability on the island without the influence of people was apparently generating this pattern through disruptive selection or divergent selection, which increases the variance and decreases that the fitness and therefore the frequency of that intermediate beak size. And by frequency, we mean proportion of birds or number of birds. Any questions about that? Directional selection can go either way. It just happened to go that direction in the lizards. And then we've got this one, which is disruptive selection. Okay, you're going to get comments back from us on your summaries. And these questions that you have on Moodle, make sure that if you didn't address all of them, make sure they are addressed at least to some extent in your final version. And again, we've got the rubric up, so you can use that rubric. Okay. Share screen. Okay, we're going to talk about inheritance. So we're going to switch gears back to genetics and the stuff we were talking about before the exam. And then we're going to take that forward into understanding Mendel and how genetic information is passed on from one generation to another. So first of all, I want to make sure we know the difference between heritability and inheritance. Then we're going to talk about Mendel's laws and this term particulate inheritance. And then we're going to get into Punnett squares and the difference between meiosis and mitosis and how that relates to variation and the premises of evolution by natural selection. It's a theme we're going to keep returning to. So you saw that on this exam that we just had. It's not going to be the last time you're going to see those premises or preconditions. So I'd like to remind you all about mutations. Last time we saw that mutations come in a variety of forms. We talked about substitution mutations, insertion or deletion, where you lose a nucleotide from the DNA sequence or add one in, or you can add or remove a set of them as well. Duplications, so as the DNA molecule is being replicated, sometimes a stretch of that DNA can be duplicated. It's like writing the same word twice in Microsoft Word, right, while you're writing your paper. Mutations, for the purposes of this class, we can think of them as happening randomly. They're just errors when you're copying the DNA. Does anyone remember what we mean by silent mutations? When is a mutation, like a substitution mutation, for example, silent?
When it doesn't affect the phenotype.
Good. When it doesn't affect the phenotype, and in the case of the mutations we talked about before the exam, usually that means you substitute one nucleotide and you get the same amino acid. And so then the protein itself doesn't change at all. It turns out that because mutations are really common, and a lot of them have little effect, they're either silent, or even if there is a little change to the protein, maybe it doesn't have a huge effect on the fitness of the organism. What that means is that we have lots of variation, polymorphism among genes as a result of mutation is really common for a lot of traits. This was something that was sort of surprising to geneticists who were first looking at genetic variation, just how common and widespread it is. But now maybe it should be obvious that there's lots, for example, lots of different eye colors and lots of different ways to get to the same eye color that don't have a whole big effect on our fitness levels. Lots of other things matter a lot more. So variation caused by mutation that happens randomly is real, and sometimes that variation has fitness consequences. When it does, that's when natural selection operates. Rates of variation that I said are random, even though they're random, it's the rates of mutation are very different in different organisms, different genes. One of the things that makes a big influence is generation time. How many generations of an organism can you pack into a given amount of time? Those with short generation times often have higher mutation rates, but it also varies among genes. So when Darwin was writing about evolution by natural selection, this stuff we didn't really know about. We didn't know about DNA. We didn't know about mutation as the source of variation. And the idea at the time was more like a blending interpretation of inheritance. You take all the traits of one parent and all the traits of the other parent, and you kind of blend them together, and something kind of intermediate comes out. that was kind of the conventional wisdom at the time, although there were lots of sort of mental gymnastics people were doing to understand why that sometimes is the case, sometimes not, right? You have a bunch of Labradors, you might breed them together, a golden lab and a black lab. Well, you don't get some kind of like dark golden lab. You tend to get yellow labs and black labs and brown labs. So how is that intermediate? Well, people invented all kinds of stories for that. Now we understand it, and it's largely thanks to Gregor Mendel, who was interested in genetics, and he did a whole series of experiments while working in an abbey in Czechia, I think that was in Czech Republic, and he used peas as a model organism. There are several organisms that are used as model organisms for biology today, like fruit flies and Arabidopsis, which is a little plant in the mustard family. We use C. elegans and its close relatives here at Jewel. But Mendel used peas as his model organism for understanding how traits are passed on from one generation to another. There's a few reasons he used peas. He used pea plants because they're easy to grow and they have short generation times, so he could do a lot of crosses and a lot of experiments. Another reason he used them is that they are not self-incompatible. What plant do you guys know about that is self-incompatible?
Goldenrod.
Yeah, so that would not have been a good plant for Mendel to use. He used peas, which meant he could take the pollen off of the anthers and put it directly onto the stigma, and it would fertilize the eggs inside those ovules. So he would know exactly who the parents are of each seed. This means through repeated selfing, if you take a plant and you self it, and then you self it again, you can get true breeding plants, meaning that the traits of the descendants are the same generation after generation after generation. That's what it means to breed true. Now, Mendel was able to create true breeding lines or lineages of peas for a variety of different traits. So every generation he would get the same tall plants. Every generation you get the same yellow peas out of the pea pod. And he could do that for each of these traits. And he found that there were some interesting patterns like dominance and recessive that we'll talk about in just a moment. So he tried all these different crosses after creating those true breeding lines and came up with a certain set of conclusions. He found that each trait is determined by a pair of genes in the plant. So I'm going to stop sharing for just a moment since we're almost done. He found that each gene was controlled by each trait is controlled by a pair of genes, which is also the case for humans. Fortunately, the peas are more similar to us in that way. But that often we had this pattern of dominance. Let's see, what do I have here? Which Mendel called his law of dominance, which just meant that if you had a gene for a yellow pea and a gene for a green pea in a single plant, then the plant was always going to produce yellow peas. If you had a gene for green and another gene for green, then that plant was going to actually produce green peas. And then if you had yellow and yellow, you're going to get yellow. But if you have yellow and green, you always get yellow. The yellow is dominant. So Mendel's law of dominance is that there's a pair of genes for each trait. One of them is dominant and one of them is recessive, such that if you have one of each, you express the dominant phenotype. Does that make sense? He also concluded that when the plant makes gametes, when it makes the sperm and the egg cells, each gamete contains only one version of the gene. So if you have a gene for yellow and a gene for green, each sperm cell or each egg cell either gets the yellow or the green, doesn't get both. Because you have to have half the number of genes so that when you meet the other gamete, you get a full set, you get a next generation that works. So this, what he called was the law of segregation. Genes are randomly separated into gametes such that each gamete has one copy of each gene. I keep talking about these different genes. We talked about yellow and green as an example. A version of a gene, so a gene that controls color, could have two versions, yellow or green, is called an allele. So this term allele, version of a gene, is something you want to make sure you know. Vocab word to know. Because organisms have two copies of each gene, when they make gametes, those pairs are segregated into the gametes. That's Mendel's law of segregation. And finally, he deduced the law of independent assortment, which means that you have many genes, right? There's all these different genes for different traits, like color and height and wrinkled or round, what color the seed coat is, white or green, lots of different traits here. Each one of those separates its pair of alleles into the gametes independently of each other. So that was his law of independent assortment. And that one will become clear in just a little bit. Maybe we'll get to that next time. Let's think about a single trait. And we're going to use a tool developed several decades after Mendel's experiments by a guy named Reginald Punnett. And so we use what are called Punnett squares. So we'll use the Punnett square to think through how we do a cross. So imagine we've got a true breeding line of peas that produce round seeds. So when you open the pea pod up, the peas are the seeds, and you've got a true breeding line where they're always around, generation after generation, when you self them. And another one that has wrinkled seeds. You can ignore the green color for now. It's just round or wrinkled. Now, the law of segregation means that you have to create gametes. So big R is the genotype, another vocab word. It's the genotype of that top parent. And little r is the genotype of the second parent. The genes are segregated independently into the gametes. So that means for each of these parents, there are two possible gametes. You can either get the big R on the left, or you get the big R on the right for each gamete. And then for the bottom one, you can either get the little r on the left or the little r on the right. The result's the same, but you have to do that step of thinking about how each gamete only gets one copy. Then Mendel crossed these. If you cross a big R, and a little r, all of the offspring are going to get a big R from one parent and a little r from the other parent. So they all get this genotype, big R, little r. And because of the law of dominance, that genotype produces a phenotype with round seeds every time. But if you take these two, or take these offspring, and you cross the siblings through self-pollination, you get the setup for a Punnett square. Once again, there are two possible gametes, either a big R or a little r, and there are two parents. So the left side of the square is one parent and it's two gamete possibilities. And then the top of the square is the other parent and it's two gamete possibilities. Then you get a grid of four possible combinations of those four gametes. When you put those two together, you get a complete set, right? You get two copies of each gene. But which allele you get depends on which row and which column you're looking at. So you get 3/4 or about 75% of all the offspring from this self-pollination cross are going to have round seeds. Some of them are going to have a big R and a little r, we call heterozygous, different zygotes, heterozygous. Some of them are going to have big R, homozygous for the dominant trait, homozygous dominant, big R, but they're all round. And then the other quarter, the other 25%, get two little r's, homozygous recessive. We can think about those percentages as ratios as well. Maybe that's a little easier. 3 to 1 are going to have this round versus wrinkled phenotype. But remember, not all the round ones have the same genotype. So you get a ratio there. One big R, 2 big R, little r, and 1 little r. That's the basics of a Punnett square. If you can create the gametes and put them on the side and top of the square, it's pretty easy to put them together. But make sure you know about the law of dominance that tells you what the phenotype is, and you know about how to separate out the gametes, make those gametes so that you can set up the Punnett square correctly. We're going to do some Punnett square practice stuff after we talk about cell division next time. That's it for today, and I will see you guys on Wednesday. And we'll have some more time for questions.
Dr. Judd,
Yeah.
what do we need to have prepared for our lab tomorrow?
You guys should have the getting to know goldenrods sheet. So you should know about the structure of a flower versus the goldenrod structure. And you guys should have decent answers to the data management questions.
Okay, but we didn't need to go out and make any observations yet, did we?
We're going to give you protocols this week, so you'll be able to jump right into that.
Sounds good. Thank you.
We have finished putting them together between now and tomorrow morning.
Right.
But yeah,
Okay.
hopefully we'll have protocols and videos of us doing things in the field, and yeah, so we're just putting things together.
Okay, thank you.
Hey Nathan.
Vocab list, yeah.
I'm loading the exam parts into emails. When I get finished with all of them, I will ship them off to the students. I'm saving everything as drafts right now. Can you enter the exam scores into Moodle at some point?
I did that right before class.
Oh, did you? Okay, so you got them in. I didn't go and look. So, and I sent everybody an email basically explaining that you're going to get an email from me with both pieces of your test. Told them how it's calculated in there, and they should check and make sure that the numbers that they see are the numbers that they see on Moodle. So.
Okay. Yeah, I don't think I had any leftover questions from that. I think I sent you an email saying that I entered them, but I didn't have time to make a histogram or anything.
Okay. All right. Cool. Maybe we'll address it in more detail on Wednesday.
Yeah, that's fine. Yeah.
Because they'll have them all back at that point.
Yeah.
I'm up to Alexis Harper. I'm slowly working my way through.
Yeah.
Down the list.
Right.
All right. See ya. Have a good ecology class.
Sure. See you soon.
Yeah. Bye.